Hello, magnificent students. This is Heidi, your instructor, and I'm going to teach you about the structure of DNA. DNA stands for deoxyribonucleic acid, and it is made up of nucleotides. Each nucleotide consists of 3 different parts. First, there is the sugar, which is deoxyribose. Then there is a phosphate. Then there is also a nitrogen base. This nitrogen base can be adenine, can be thymine, it can be cytosine, or it can be guanine. This is one nucleotide. DNA consists of a series of nucleotides that are covalently bonded between the phosphates and the sugars. So you have one nucleotide with another nucleotide bonded to it, another nucleotide bonded to that, and it continues in a series such as you see here. So we have a backbone that consists of sugar, phosphate alternating, covalently bonded, so it's very strong. This backbone isn't going to break. The middle, though, is going to be consisting of nucleic acid bases that are hydrogen bonded to other bases. So adenine is going to be bonded to thymine, and guanine is going to be bonded to cytosine. Now, each of these bases that are bonded to the base in this one are also bonded to a sugar and a phosphate on their end. So this thymine is bonded to an adenine, which has its own sugar and phosphate. This cytosine is bonded to a guanine, which also has its own sugar and phosphate, and its sugar is going to be bound to the phosphates of the previous nucleic acid. Then its phosphate will be bound to the sugar of this nucleic acid that has the thymine. Then this guanine is going to be bound to cytosine. This guanine is bound to cytosine. And we have another guanine that is bound to cytosine. So here's a section of a DNA molecule, and I want you to notice a few things. First of all, the backbone is made of phosphate sugar on both sides. These are covalently bonded so they don't break apart. The center, or the rungs of the ladder, are going to be made by the nucleic acid bases, and guanine always pairs with cytosine, adenine always pairs with thymine. These are hydrogen bonded so that it is easy for them to break apart. That will become important later on in what we learn. I also want you to notice that it is running what we call anti-parallel, meaning that this strand is going up while this strand is going down. It's kind of hard to see on this camera, but if you look a little bit closer, you can see how one strand is right side up and the other strand is upside down. We call this running anti-parallel. The way that we determine that this is anti-parallel is we're going to identify one end as being the 5' end and the other end as being the 3' end. So one strand is going to run from the 5' to 3' direction, while the other one runs from the 3' to 5' direction. That is anti-parallel. We say that the structure of DNA is a double helix. It's double because there are 2 strands, and it's a helix because it actually twists like this, making a double helix form. DNA is a very long molecule that codes for all of the proteins in your body. A section of DNA that codes for a specific protein is called a gene. Because it's so long, DNA has to be packaged into tight structures that are called chromosomes. Chromosomes stay in the nucleus of the cell.